urea cycle is a biological pathway that converts the toxic ammonia into less toxic and water-soluble form urea that can be secreted out as urine. Along with that, urea cycle is also important for recycling the intermediates of the citric acid cycle and the production of arginine. Why is there a need to convert ammonia into urea? Let's understand it in the context of the body's most pivotal tissue, brain tissue. Ammonia is a neurotoxin. It is rapidly diffused into the brain tissue. The astrocytes, the most abundant glial cells in the brain, are responsible for converting the toxic ammonia into the glutamine and convert glutamine into glutamate. This hence disturbs the glutamine-glutamate cycle. In normal functional synapse, glutamate is the primary excitatory neurotransmitter, which is a chemical released in the neural synapse to help transmit the nerve impulse from presynaptic neuron to postsynaptic neuron in brain. Excessive glutamate binds to NMDA receptors, leading to an influx of calcium and sodium ions into the neuron. The influx of calcium activates nitrogen oxide synthase. Nitrogen oxide synthase catalyzes the formation of peroxynitrite from nitrogen oxide and peroxide. The peroxynitrite disrupts mitochondrial function and triggers the release of reactive oxygen species, ROS, and result in lower ATP. Depending on the severity and duration of the excitotoxic insult, neurons can die through apoptosis, programmed cell death, or necrosis, cell death due to injury. The urea cycle and the TCA cycle are two essential metabolic pathways, and they're more connected than you think. But the urea cycle produces fumarate recycled into the TCA cycle, where it helps generate energy through ATP production. Meanwhile, the TCA cycle produces oxaloacetate, which can be converted into aspartate, an amino donor in the urea cycle. This exchange, fumarate to TCA, aspartate to urea, creates a metabolic bridge, linking energy production with nitrogen disposal. Together, the urea and TCA cycles recycle key intermediates, keeping your cells energized and ammonia-free. Going back to the formation of the fumarate, arginosuccinate, an intermediate of urea cycle split into fumarate, which goes to the TCA cycle, and arginine. Arginine can be used for protein synthesis, nitric oxide production, or continue in the cycle, where it's cleaved by arginase to release urea and regenerate ornithine. So, while arginine is a non-essential amino acid, your body works hard to make it in urea cycle. Hyperammonemia occurs when there's an excess of ammonia in the blood. Normally, the liver converts it into urea for excretion. But when the liver is damaged, or a genetic disorder is present, ammonia builds up, causing harm. The two main causes are liver disease and urea cycle disorders. In liver disease, ammonia isn't processed properly. In urea cycle disorders, the enzymes that convert ammonia into urea are faulty. Symptoms range from mild nausea and pain, to severe confusion, seizures, and even coma if untreated. Diagnosis includes blood ammonia testing, liver function analysis, and brain imaging to check for damage. Treatment focuses on lowering ammonia levels, by stopping protein intake, giving glucose solution, or using dialysis. Genetic disorders affecting urea cycle are major cause of inherited hyperammonemia. Type 1 is caused by a defect in the CPS1 enzyme. It's autosomal recessive and doesn't involve orotic aciduria. Type 1 can lead to brain swelling, coma, and even death if untreated. Type 2 is the most common form. It's X-linked recessive and involves a defect in the OTC enzyme. Orotic aciduria is present here. That's all about significance of urea cycle and its disorder, hyperammonemia.
Please like, share and subscribe and turn the bell icon for more videos.